So good afternoon, everyone. I think I'm fortunate to be the last speaker, not because the empty chairs, but because it's like an introduction to what I'm going to talk about. It's a pilot study, but I think it touches with many of the aspects that were brought before. The aim of the study was based on the first understanding that, as was talked so many times before, that the prevalence of autism is increasing dramatically in the last 2 decades. We know that looking at the extensive genes, genetic research, we know that the heritability is high on the one hand, 60 to 90%. However, on the other side, if you look on the genetic causes, it may explain with association, not with a direct connection, about 30% of the children with autism. So what is left is to look for other causes that may be associated with genes, maybe not, that is leading to the development of autism. And one of them is that has come up is environmental effects, mainly affecting the immune system, and we'll talk today mainly on the effect during pregnancy. We know that testing there is association between causes between autoimmune disease, for example, psoriatic arthritis, or that leading to increase of IL-17 that is known to cause autism in animal models. We know that there are signs of that the viral infection may lead to not necessarily that the mother is sick, but there is activation of the immune system that eventually leads to activation of the microglia or direct effects that when we know that microglia is highly associated with brain development and may lead to development of autism eventually. So we wanted to test these questions by testing the protein composition in the plasma of cord blood. We look for proteins and not for RNA or other metabolites, although it makes sense to look there also, but we wanted to look at the end of the cascade, not the complete end of the cascade of productions of proteins. It was mentioned before about the protein modification, which is another stage, but we wanted to see whether the proteins that the profile of proteins is different between children with that eventually will develop autism and children with neurotypical children. The way that we got the sample is because it's part of studies that we do in Sheba, testing whether treatment of umbilical cord blood may have an effect on the children with autism, with the thought that cord blood is leading to immune modulation, and that would lead to improvement of the autistic symptoms. So we knew that the children has autism, and we had their plasma from their cord blood, and we compared it with neurotypical children that we got their cord blood sample from the public cord blood. These are units that were thrown because they were not, the size of the unit did not fit the cord blood bank. Specifically, we wanted to look for neuroinflammation proteins that in our hypothesis were thought to be there in the plasma of the cord blood. This is our cohort. We had 30 children with autistic, diagnosed with autism, compared with neurotypicals. All of them, the autistic children did not have any known genetic causes, and is on the neurotypical, it was, we assumed that none of them develop autism. The likelihood of autism in Israel is 1%, 1.5%. So the likelihood that one of them by chance would fit this cohort is there, but it's very, very low. Looking at epidemiology of the cohorts. So there is a difference in the sex. So the gender, the sex in the neurotypicals were 15-15, male and female, whereas as I would say, as expected in the autistic group, there was 4 times or even 4.5 times more males than female. But otherwise, the mother is age, gestational age, weight of the child at birth, Apgar score were similar between the groups. And the ways that the autistic spectrum severity as measured by ADOS and the psychologists and neurologists that diagnose these children was split by 1/3 to mild, moderate, and severe, as we find in the overall autistic population. What we were looking at is proteins that were encapsulated in microsomes, extracellular vesicles, exosomes, microsomes. There are different names for that. These are encapsulated miniature vesicles that are getting exerted from the cells. The origin is either from the endoplasmatic reticulum or from the cytoplasm. There are a lot of speculation what's the role of exosomes in the body, in the tissue. Currently, the idea is that they have a role in signaling, signaling from cell to cell the status of the cells in order to convey information if there is some changes in the tissue. And we look at it as a reflection of the cellular status. So the composition of the proteins within the exosome can give us some insight of what's the status of the proteins in the whole tissues in the body. We did not look specifically, the exosomes that we looked at in the plasma was not specific to the brain, but they presented the whole body. So what is done is we do proteomics. So we take the exosome, we take the proteins, and we run them through a centrifuge, and then in the columns, and we identify all the proteins. And this is the plot that we get. Oh, this is the initial results that we get after this procedure. What you can see on the x-axis, yes, okay. On the x-axis, you can see the difference, okay, like the mean difference between the comparison between proteins, and this is in comparison to the variance, okay. That's and everything that is in red is a significant variance between the neurotypical and autistic children. Everything that in the negative part, meaning that the expression of proteins were higher in the autistic children, whereas the other side is in the neurotypical. And clearly you can see, we'll get to the number in a minute, that even just by looking at it, you can see that there are a very large group of proteins that had higher expression in the autistic children. These are the numbers. About 5,000 proteins were expressed. In the procedure, we only took around 3,000 of them because we only took the ones that were expressed in 70% of the children. And if we split, and if we split them, so we take that about 565 proteins were expressed differently between the autistic and the neurotypical children. What do we mean by expressed differently? I had experience from previous lectures that it's difficult to understand the coming slide, so this is a private example. So we're looking here at the expression of the protein GLUT1. It was mentioned before, it transfers the glutamine to glutamate within the mitochondria. It's a mitochondrial protein. What you see on the, it's not working, what you see on the left in blue, these are, and you see the line in the middle. So everything on the left in blue, they are the samples, the neurotypical sample, and on the right, it's the autistic sample, 30 on each child, on each side. And you can see, without statistics, you can see that if I take the mean expression of the left side is lower from the right side, okay, mean expression is lower on the neurotypical. But what is more dramatic, that the variance of the expression in the autistic children is much, much lower than the neurotypical. And you can see that over here, over the 500 proteins, okay? On the left side, this is the mean, okay? The same procedure for all the 565 proteins, okay? And what is in brown is the autistic children. You can see that it's deviated to the right, meaning that higher mean expression. But again, what is really striking is that on the right side, the variance of in the different group is much, much lower for the autistic children. What it means that if you look at from a higher view, that the profile of the proteins in the autistic children group was really similar, very, very similar, because the variance was really low. So it looked for most of them like this, okay? Similar expression. Oop. In the next step, we wanted to look further to understand what's the composition of these proteins. So we took the string analysis. It's a big data analysis based on animal, human studies and manuscripts that were published. And we saw that these proteins are split into 3 groups based on subcellular organelles. The red group, which is the largest, are proteins coming from the mitochondria. It's the mitochondrial proteins. The green group, the second largest, is the endoplasmatic reticulum proteins. The blue one is a bit of a mixture. It has some of it are neuroinflammatory proteins, but some of it are cytoskeleton proteins, and so it's kind of a mixture. Trying to play with the clustering, going if someone did that going up and down, it really was much more messy than what we got here. This is the most clearest view. The next step, what we did is to ask, this plasma is coming from the whole body, so we wanted to know which are the proteins that are coming specifically from the brain. So we look at another data source, is the Human Protein Atlas, and we compared the proteins coming from the brain with our proteins, and we found out that there were 12 proteins that are dominant coming from the brain, dominantly coming from the brain within the different proteins group. These proteins are, have some roles, but most of them, oh, sorry, not most of them, some of them, the one in green, coming from the endoplasmatic reticulum, are known to be associated with endoplasmatic reticulum stress response in conditions where there is stress to the cell. So the endoplasmatic, there is abnormal folding of proteins, and the endoplasmatic reticulum has specific response to that, and these proteins are involved in this response. In the blue group, I will just mention two proteins. COPA is a known neuroinflammatory involved in many processes, neuroinflammation, and Rialin here, that I know the neurologists here known are known in neurological, known to cause epileptic encephalopathy, brain malformation, and they also associate with autism. And here is our friend GLUT1 from the beginning. So if we want to conclude the results until now, we saw that there are increased expression of proteins within the autistic children group. We know, I think the most striking one is not that the increased expression, but the variance is really, really low. And although it is a pilot study, this was really a striking result that implies that we need to further test it in the future, that maybe there is kind of a profile of a signature that is specific for autistic children. And we found that there is a specific presentation of mitochondrial endoplasmatic proteins that are, and these proteins are known to be increased in conditions where the mitochondria and the endoplasmatic reticulum are under stress. And we found that some of these proteins are also found in the brain. Diving into the literature, once we found these findings, so we found that there are quite, in the recent years, there's a lot of literature talking about the effect of, or the association between mitochondria and synaptogenesis. The rate of mitochondrial maturation and metabolic, and the metabolic rate is highly associated with synaptogenesis and brain development, and similarly to endoplasmatic reticulum. So based on this understanding, we hypothesize that maybe what we see here is it's a kind of that there is that there are many etiologies starting the process that eventually leads to abnormal brain development. It could be coming from genetic factors, could be coming from epigenetic, and everything in the middle, like spliceosome, splicing, all of these processes can lead to autism, or it could be due to infection and direct effect of the infection on microglia or on the brain development. But everything is converges into the effect on mitochondria and endoplasmatic reticulum. We tested it during the in cord blood at the time of delivery, but it also can happen before. And potentially the abnormal activity of mitochondria and endoplasmatic reticulum eventually leads to abnormal synaptogenesis and myelination and may lead to autism. Clearly, this hypothesis needs to be tested. So our future plans are few. One is to repeat the study and to do it on a larger group, and to test it not only on exosome rising from the whole body, but to use antigens to split the brain-derived exosome from the rest of the exosome, and to look into the content of these exosomes. And what is done currently is to grow iPSCs coming from the children, the autistic children in this group. And this is the first colony of this IPS, and to look in the future whether we see abnormal mitochondrial function or endoplasmatic function once it will mature into will mature it or will direct it to develop to be a neuron. This is on one side. On the other side, if we want to use the concepts that that EVs and the exosomes present or reflects the protein profile of the developing brain, is to look into to sample fetuses during pregnancy, testing it from either maternal mother blood samples or from doing amniocentesis from the amniotic fluid, and to test the children in blood samples in the first 2 years of life, during the times that we know that synaptogenesis and afterwards pruning and myelination is very extensive, and to see whether there is this profile and the difference between autism and neurotypical children continues during these 2 years of life. So, just to, in the conclusion of the conclusions, to thank the group of great women that working with me and researchers, and thank you all.